Now talk about the role of gene ontology in solving these problems. And so hopefully that gives you an idea, like a kind of a vague idea of, what exactly is the role of gene set enrichment analysis in terms of things like a CRISPR screen. So really the goal here again is to take a set of genes which can look random at first glance, and then basically just ask the question, okay, are there common pathways that these genes are pointing to that may be responsible for, in this case, drug sensitivity of cancer cells? And so, the general question is, I do run a screen and I get some X number of genes, like 100 genes that are associated with some phenotype of interest. And we're just asking the question, what do these genes do? And do they point to some key pathway or function? And so one of the main problems with asking this question is, if you want to, in an unbiased way, look at a set of genes out of some CRISPR screen and ask the question, are these genes involved in some pathway or function? You of course need some kind of database to tell you, okay, well, this gene is previously characterized as performing, function X or function Y and so on. And so you need some kind of way of looking at literature and on gene function and, querying this literature to say, okay, what does each gene in my gene list do? Right. And so there's multiple problems with storing the literature in a so-called computable way. But one of the main problems is that of inconsistency, is that, multiple people can use the same name for different concepts, or, there can be multiple names that refer to the same concept. Right. And so here's a silly example where, if you think about the word cell can refer to two very different concepts, either like an actual cell or like a prison cell. So in biology, the problem of coming up with the same name for the same concepts is very challenging. Even at the level of naming genes, there's a lot of disagreement and discrepancy in the literature. So here, for example, is information on one particular transcription factor, HNF4 alpha. And, basically at the bottom of this slide lists a bunch of aliases and synonyms for HNF4 alpha. And HNF4 alpha is a fairly important hepatic transcription factor that is pretty well characterized. And so you would expect for such a well-studied gene that we could all, come up with one consistent name for this gene. But this webpage from GeneCards just gives you an illustration as to how many different ways this gene has been typically referred to in the literature. And not all of them are obvious. So you can see synonyms like NR2A21 or MODI, for example, are used to refer to HNF4 alpha. And so if we can't even agree on common naming schemes for genes, then you could imagine that there's just, a ton of different ways of referring to the same concepts when it comes to like gene function or pathway or cellular function or things like this. And so the point here is that coming up with a consistent naming scheme that everybody agrees on and everybody uses in order to kind of store and retrieve information about gene function and things like this is very challenging. So there's two common approaches to dealing with the issue of naming, one of which is called controlled vocabularies. So a controlled vocabulary is basically essentially like having a drop-down list for people to select the names of functions that genes might carry out in our case. And so basically controlled vocabularies is just a set of restricted terms that people are allowed to use in order to describe gene function. And so this definitely gets around the problem of coming up with a common naming scheme. I mean, there's other problems that it has, namely that lots of people can come up with controlled vocabularies, for example, for gene function, but the main challenge is having people agree to using it. And so in practice, for a lot of controlled vocabularies, you have to have like big organizational bodies like the gene ontology that we'll be talking about soon. They get together and try to come to some consensus about what names should be used for gene function, and tries to convince everyone to use it. But one problem with controlled vocabularies is solved by ontologies. And so the idea of an ontology is that ontologies are kind of like controlled vocabularies on steroids. So ontologies allow you to it allows you to describe relationships between concepts. And so one thing that, for example, a controlled vocabulary cannot do is that it cannot encode the fact that the S phase is one part of a cell cycle. And so the concepts of cell cycle and S phase are completely separate, and there's no way to encode that one is related to the other. But in an ontology, ontologies basically give you a mechanism for describing those kind of like part of relationships. So gene ontology is an ontology that's principally concerned with the function of genes. Within gene ontology, the goal is to categorize different kinds of annotations under three broad core terms. And so the gene ontology is principally concerned with characterizing the molecular function of genes. So very kind of recording elementary activities or tasks, like whether a gene product is like a protein kinase or insulator or insulin receptor. They're also concerned with kind of more coarser resolution biological processes. So things like, are you involved in the cell cycle process or apoptosis? And finally, they're concerned with where gene products get localized. And so, for example, within the mitochondria or within the nucleus or different compartments of the cell. So underneath each one of these three core terms, the molecular function, biological process, and cellular component, Go is basically a hierarchy of terms, or more formally, what's known as a directed acyclic graph. And so the idea is that with the exception of the three core terms, every annotation within gene ontology has some kind of parent. And the idea is that at the top of this hierarchy are the kind of more broad terms. So at the very top of, each one of the three hierarchies is one of the core terms, in this case, biological process. And as you move kind of down in this hierarchy, you get more and more specific terms. And so the idea is that genes can be annotated with one or more terms. And when a gene is annotated with a very specific term, like say, if you look at one of the terms at the very bottom, so like positive regulation of signal transduction, then if a gene is annotated with that term, then it's automatically annotated typically with all of the terms above it. Because the idea is that if you are annotated with a very specific function, then all of the parent categories of that function should also apply to the same gene.